So good afternoon, everyone, and thank you so much for this invitation to speak. It's a real honor to be here. It's an honor to speak in a series that has seen so many well-established forest scientists and people in the forest industry come through. So I really must thank the Department of Renewable Resources, Nadir, Andreas, Vic, Annette, and the other staff for having me here today. And thank you to you for braving the storm out there. So, can genetics help climate-proof our forests? I wanted to be provocative, but if I wanted to be really provocative, I would have said, can genetics climate-proof our forests? And then I could have stood up here and said, no. And I could have left, you could have left, and we could have started the banquet early. And as I stepped out of the Lister Center, where I'm staying this morning, thinking I might go for a run, discovering the freezing rain and the treacherous conditions, and thinking a trip to the emergency room probably wouldn't be a good start to my lecture, I was thinking it might be quite hard to convince you that we need to worry about climate-proofing our forests when things in Edmonton seem much the same as always, at least today. And I am an Albertan, but I won't tell you where I'm from. So you can probably guess. So I think, without a doubt, the world is warming. And if you'd like to know more about recent analysis of the warming that's already happened to date, I refer you to this Berkeley Earth project, which has reanalyzed climate data from around the world. The analytical team comprised people who have not analyzed these sorts of data before, but they were statisticians, very well-regarded statisticians, and some of them were climate skeptics. And the results that they came up with were pretty much identical to those of the IPCC and other analyses. And so without a doubt, things are getting warmer. Those effects have been already seen on forests globally, and Andreas Hammond here has done a lot of work that has shown already effects on forests and how much the climate in Western Canada has already changed. So these are maps. Here's a map of 25-year trend in precipitation, 25-year trend in winter temperature. And what we see is that things are generally getting warmer. Also, in Alberta, things are getting drier. In British Columbia, it depends where you are, what the effect on precipitation has been. But these changes in climate can be directly or are indirectly or directly implicated in various changes in forest health, including the mountain pine beetle, the spruce and aspen dieback that you have seen in Alberta and that the Canadian Forest Service has done much work on, and things like the fungus Dothostroma outbreak on lodgepole pine in British Columbia on the coast. So, what does this mean for our tree species in natural populations, and what does it mean for the trees that we stick in the ground? Well, this cartoon depicts a species range along some climatic gradient. So you might think of this from being south to north, for example, and it goes from warm to cold. This is the historical range of the species. We've got perhaps a few disjunct populations at the southern margin or the warm margin. We might have a few up here too. I haven't drawn those in. And that is the space, and that reflects the climate over which that species can survive, establish, grow, and reproduce. And what we have in a warming climate is we have a shifting climatic niche. Again, for one species. So we have some overlap between the old range and the new range, probably a fair amount, the new potential range, but we have some changes in that. So for example, we have populations at the warm margin that might no longer be within that climatic niche of the species. We don't exactly know what's going to happen to those populations, but we know they're not located in conditions where we have found them naturally. We have this portion of the range that overlaps between the historical and the current, but the conditions have changed there. So we still have the species there, but we may not have the right trees of that species in that location. And finally, at the cool margin, we have this habitat potentially becoming available that the species could move into. Now, for a moment, I want to concentrate on this area of overlap, because generally for most of our species, that's where a lot of the forestry happens, is in the relative core portion of the species range, where that species does well, competes well against others, and produces well. So let's just think about this overlapping area. And here's a schematic showing the distribution of trees, again, within a single species, distributed over a gradient. And what we expect to see is we expect to see trees adapted to warmer conditions in the warmer portions of that zone, trees adapted to moderate conditions in the middle, and trees adapted more to cold conditions in the north. And there's overlap where you see those genotypes. And of course, it's a continuum. I've drawn 3 colors, but it's a continuum of adaptation. And so what we see then, what we expect to see with climate warming, is that the populations become mismatched. The colors no longer match with the background as this whole area has warmed up. The gray here depicts trees that perhaps are becoming unhealthy, perhaps are dying, perhaps aren't reproducing. And this is the problem that we face when we're making those reforestation decisions in areas where we're still planting the species that are already present, but we've got to start thinking about what we're planting. Now, there's no doubt that trees, individual trees, can be very resistant to changes and can persist for a very long time. Here is a Norway spruce in Sweden that's estimated to be 9,000 years old. Now, it's not much of a tree. It's only a few meters tall. But the fact is that it has survived huge fluctuations in temperature since the last ice age, and it has persisted. Now, it keeps sending, it's growing in Krumholtz form and keeps sending up new shoots over time. But a very old individual, certainly not very productive and not a forest. But trees may be able to hang on quite a long time, but they may not be very productive, they may not be very healthy, or we may see the sorts of diebacks we've seen in lots of species. So what I'm going to talk about today is as follows. First of all, I'm going to talk about what we've learned about genetics and climate from provenance trials, from common garden experiments, looking at traits of trees that are grown from different seed sources. And then I'll talk about how we can use genomic methods to get at that genetic basis of adaptation to climate, what really matters. And third part of the talk, I'll talk about the capacity for natural populations to migrate, to track their climates, or to adapt in place to new conditions. The fourth topic is chasing a moving target. As the climate warms, how do we get the right trees in the ground in the right place at the right time? And finally, I'll talk about issues around assisted migration, which is the movement of organisms, in this case of trees, to help them track their optimal climates. So, the traditional approach to studying genetics and climate. And here we're talking about studying genetic differences within the historical range of a species. Now, we have to think like a tree for a minute here. What do trees have to do to be adapted to their environment? First of all, they've got to start growing after the risk of hard frosts is done, after the risk of damaging temperatures is done in the spring. But they need to get out of the gate fast after that danger passes. They need to grow quickly enough to compete against other trees of their species and other species for light. And they need to cease growth, set bud, and develop cold hardiness before low temperatures arrive in the fall. Now, we've done quite a bit of work with Sitka spruce as kind of a model to work on around climatic adaptation and other factors. It's got a very convenient range, a long, narrow range that extends from Northern California, where it persisted at the last ice age, up the coast to Kodiak Island, Alaska, which had only reached about less than 1,000 years ago, probably about 500 years ago. And when we take trees from California populations, and we take, so warm adapted trees and cold adapted trees from Alaska. The trees from California grow very rapidly in Vancouver, and they grow until about November or even early December, and they get hit by our frosts, which tend to come quite late in Vancouver. The trees from Alaska, they grow, they break bud, they all break bud about the same time in the spring. They grow a little bit, and then they hunker down. Winter's coming, they're adapted to a very short growing season, tremendously cold hardy, but they don't grow after about the end of July. And these are seedlings. So if we take trees from different locations all along that range, from California to Alaska, and we arrange them by the mean annual temperature of their source, which is highly correlated with latitude, 17 populations along here, and we look at the timing of bud set. So this is the day of the year that they set bud, seedlings, and this is the height growth that they achieved in this common garden. We see that both the timing of bud set and the amount of growth they achieve are very much a function of where they came from. Very tight correlations here with both bud set and with growth, reflecting that historical adaptation to those conditions. Now, if we look at this right in the middle of the range, this is Prince Rupert, where that triangle is. And if we look, Prince Rupert's got a mean annual temperature of about 7 degrees. And it sets bud here on about the 215th day of the year. If we take projected warming, middle of the road scenario for warming, that corresponds to 10 to 11 degrees mean annual temperature. And we expect trees adapted to that temperature would grow for another 50 or 60 days or more. So what this shows is the shift in synchronization. We expect to see the climates shifted, but the trees will still be growing according to their old environment. Now, Andreas and I both have worked over the years with Tong-Li Wang, who's still in my group, and done various sorts of analyses. Tong-Li has analyzed a lot of provenance trial data from a huge lodgepole pine provenance trial in BC called the Illingworth trial. 120 test sites, 120 populations, provenances sampled, seed taken from 120 locations, planted on 60 test sites. Hugely expensive experiment. It's grown for 35 years. And these are the kind of results you can get from a long-term provenance trial, just absolutely invaluable. So this experiment has multiple sites, but these are photos of trees at one site, 4 different provenances. This is a high elevation provenance from the south, doesn't grow too much. This is a provenance from the BC-Yukon border, from way up north. And these are a couple of more local. This is a relatively local provenance, and then this is a southern provenance that grows very well. And you can see the kind of variation you get. What we can do with multiple test sites is we can get these response curves where each of these lines reflects the growth of a different provenance. So trees from a different location, and we can infer their response to different climatic conditions. So if you grow them this tells you what happens if you grow them at colder temperatures, or projects what will happen if you grow them at warmer temperatures. And what we see here is that trees that come from cold locations generally have lower optimal temperatures for growth. Trees that come from warmer locations have considerably higher optimal temperatures for growth. So we don't have 35 years. And we don't have millions of dollars to put in these tests for all provenances, for all locations, and we need to think about how we're going to do this quickly. Now, lodgepole pine, we've got great data for it, but there are a lot of species that we don't have that information for, and there are areas that we don't understand what their response curves look like. So is there a way that we can look at genetic variation directly at the DNA level to understand what underlies those differences in traits? And again, we're still talking about using the historical range and adaptation across the historical range to harness that. So now I'm going to subject you to Genomics 101, which is a very what I hope is a simple explanation of how we use this kind of information to understand adaptation to climate. There's information about discovering genes for this disease and genes for that disease all over the newspaper, but what does it really mean, and how do we try to associate then changes in DNA sequence with changes in traits? So here we have, for a given gene, this is just a gene, an example, and we have a sequence for that gene, and we have two different variants, two different alleles, just like you have different alleles for eye colors. Here we have one allele where in this particular location in the genetic code we have an A. Remember, there's four nucleotides in the DNA sequence. And here we have a mutation. We've got a G in some copies instead of an A. And what people initially started looking for was these single base pair changes called single nucleotide polymorphisms. I'm going to call them SNPs through the rest of the talk. Originally, what people looked for was SNPs that had very large effects. Wouldn't it be great if we could find this single nucleotide polymorphism, and if we could find a tree that had two A's there, both copies had A, these are big trees. And boy, the G's, we don't want those. Those are little trees. And early on, molecular biologists went looking for these. We didn't find these kind of big effects. We find little effects, but nonetheless, we can find effects of these single changes in the genetic code on traits. So how can we use that with climate? Well, hopefully you all get out to the Rockies regularly. And here's a cartoon showing what we might expect to see for a given SNP that changes as you go up in elevation. Temperatures get colder, precipitation often goes up, snowpack, etc. So if we go out and we look at a mountain population up a big elevational gradient of a single species, and we see a whole bunch of these G alleles at the bottom, and we see a mix in the middle and a lot of individuals that are heterozygotes, a G and an A, and we go up high and we see that we see a lot of trees that have two copies of a G allele. That suggests, doesn't prove, but that suggests it might be involved in adaptation. Now with Sitka spruce, we've done this with that big latitudinal gradient that I told you about. And in a relatively small scale genomics effort where we looked at about 200 genes and about 650 single nucleotide polymorphisms, we found about 35 SNPs that were involved in adaptation. But of those, here are 9 that showed significant latitudinal declines. So where we see that the frequency of the cold adapted SNP, each of these is a single base pair change, a SNP in a single gene. So these abbreviations stand for the gene that those changes are in. Some of these are in the same gene, like those two. And we see this climb, these consistent climb, where the more cold adapted allele increases as you go north. And that gives us pretty good evidence of adaptation. Interestingly enough, we looked at some of these genes in mature, this was with seedlings, we looked at some of these genes in mature populations, and we found the declines for many of them were even steeper than for the seedlings, and that makes sense because the mature trees have been through selection. Now, if you can find the same climb in multiple places, go up a bunch of different mountains, or have different latitudinal transects in different geographic areas, and find patterns holding up, that's even better information that this pattern reflects adaptation and not just some historical event around colonization patterns, population history, etc. Now, the third way that we can look for adaptive variation is we can grow seedlings and phenotype them. So what I've talked about so far is looking at correlations between the genes and the climates that the trees come from, but not looking at the traits that are important. The third way is we can look at the traits and the genes, and we can look for association, statistical relationships between them. So this is called association mapping or association testing. And in this case, what we're looking for is differences in phenotypes, in traits that correspond to differences in genotypes. So here, I've cartooned what we might see. We've got individuals can have one of 3 genotypes for this SNP. They can have 2 A's, they can have an A and a G, or 2 G's. And if you see differences in the trait associated with that, then after we adjust for population differences, then that is another indicator that you've got a genetic variation that's involved in adaptation to climate. And here's an example, again, from Sitka spruce. So this is for a single gene. Again, we found 35 genes that showed we found 35 SNPs that showed associations between which SNP variant you had, which nucleotide you had, and between either the timing of bud set or cold hardiness. Now, timing of bud set and cold hardiness are the traits that show us the strongest difference among populations for most conifers that have been studied over wide areas. And so we're able to start getting at those genetic differences that collectively mean that trees set bud at different times or develop different levels of cold hardiness. And you can do this for drought hardiness, you can do this for growth rate, you can do it for wood density. But since we're interested in adaptation to climate, we're not working on things like wood density. So here's an example. This peroxidase 6 gene in one location can have either an A or a G. There are also C's and T's. I just seem to have used A's and G's throughout this presentation. And so here you see trees that had 2 G's set bud around the 200th day of the year on average, and ones that had 2 A's set bud around the 270th day. And that's a big difference. Now, this is the largest effect one we found, but there are many genes contributing, and each of those SNPs explain 1 to 5% of the variation. So that's how we get at this. I'm talking about tens of genes here, and later I'll be talking about many more than that. But this is how we go through these analyses, and we can go out to natural populations, sample the trees, and grow them up and look for these kinds of effects that are associated between genotypes and phenotypes. We can also use these SNPs, and there are millions of them in the genome. There are hundreds of thousands of them within genes. And we can use those collectively. We can use a set of SNPs to understand population structure. And this is a bit of a deviation, but this is showing how we can use data from SNPs to understand hybrid structure and how hybrid zones are structured along climatic gradients. So this is for the hybrid zone between Sitka spruce and white spruce. So in each of these graphs, if a tree's up here, it's pure Sitka spruce. If it's down at 0, it's pure white spruce. And the x-axis is showing variation in the proportion of Sitka or white spruce as you move along a gradient. So this first graph is as you move up rivers from the coast. Sitka spruce is on the coast, white spruce inland. And as you move up big rivers like the Skeena River or the Nass River, what you find is you've got pure Sitka until some distance inland, and then the proportion of white spruce starts increasing and Sitka drops off. We can see a decline with mean annual temperature. Again, as you move into colder environments, you have more white spruce. And this one's interesting. I'm talking a lot about temperature today, but for this hybrid zone, the proportion of Sitka spruce stays high with respect to precipitation until you get below about 1,000 millimeters of precipitation. And then we see a very, very steep drop in this hybrid composition, with white spruce occupying more low precipitation environments. This work was done by Jill Hamilton, who just finished her PhD, and she's now a postdoc at U of A in Janice Cook's lab in biosciences. We can also use these kinds of markers to understand the effects of selective breeding. We have breeding programs for our major tree species, and we can understand how we're changing the genetic composition of those populations through our selection for increased volume growth, changes in wood quality, etc. Here's an example in the Engelmann spruce, white spruce hybrid zone, which comes into Alberta and which is very economically important in BC. And so, and this work was done by Amanda Delatorre, another PhD student. So on this axis, we've got here's hybrid index, pure white spruce here, pure Engelmann spruce here. We've got 2 different breeding zones, and this is the breeding value for growth rate. What's the genetic quality of the individual as a parent to contribute to the next generation, to contribute to fast-growing trees? Well, 0 is an average breeding value, and you want to be selecting the trees, of course, with the positive breeding values. Well, what's interesting here, this is determined by SNPs, the hybrid index, and what you can see in both of these breeding zones in BC is that selection for faster growth rate is actually selection for more white spruce content. So the genetic effect is shifting within this hybrid towards more white spruce. Well, that's interesting with respect to a warming climate because things are getting warmer, which white spruce is more adapted to than Engelmann spruce. And in a lot of areas, things are getting drier, and white spruce also handles low moisture better than Engelmann spruce. So the breeding program, so the dots in red are the parent trees that have been selected, or the families that have been selected, and the selection, the selective breeding is helping pre-adapt those materials in this case to warmer conditions, probably partly because they've already been tested under somewhat warmer conditions than their ancestors grew under. Okay, so we've had Genomics 101, and we've had the retro approach. Let's move on to, well, maybe we don't need to do anything. What's going to happen to our populations if we just let nature take its course? And here I'm not talking so much about what we plant. I'm talking about what's going to happen to natural populations, naturally regenerating populations. So I thought about calling this talk "The Ents Are Marching," but I used that title at the University of Idaho, and it turned out that a bunch of the audience didn't know what an ent was. You would have known, but anyways. So the ents are marching. To land of gloom with tramp of doom, with roll of drum, we come, we come. Seems kind of appropriate. The trees have to move. And of course, species can migrate. They have migrated from south of the ice sheet to cover huge areas since the last ice age. But the reality is, this line here is the rate at which we expect tree species in North America, on average, to be able to move. So it's really, really slow. About 100 meters a year was the maximum post-glacial migration rate on average, based on some work done by Jason McLaughlin. The climatic envelopes of our species are moving at 7 to 10 kilometers per year on average. So the trees are going to move, but the climates are going to move a whole lot faster. Now that is based on seed. Seed has to move for the species to migrate. But we also have gene flow. We have the movement of genes via pollen. And a lot of our conifers have pollen that can move really, really long distances. This is lodgepole pine pollen. It's got a couple of air sacs like Mickey Mouse ears. And it can go up into the atmosphere and move a very long ways. Now, it's got to come down somewhere where there's individuals of the same species that are receptive, that are at the right stage of timing, and various other events have to happen. But nonetheless, pollen can travel a very long ways. Now, a little bit of pollen travels a really long ways. So from any given location, most of it is going to fall relatively close. But some of it will continue on. And this can certainly help, because this spreads genetic variation around, and it spreads, in all likelihood, genes around among different environments, different climatic conditions. But the effects of that movement are really going to depend on where the pollen comes from and where the population is that receives it. So this is some work, again, we're back to Sitka spruce, but these are estimates of gene flow rates in Sitka spruce. And this will be cumulative, this will be sort of historical gene flow levels based on what the genetic data tell us on SNPs again. So the arrows, the width of the arrows tells you how much gene flow there is between pairs of populations. And over the central part of the range, gene flow is about the same north and south. But when you get out to the edges, gene flow from the middle out is a lot more than gene flow from the edges in. And there's a lot of good theoretical reasons why that's the case. But if we look at the effect here, well, that asymmetrical gene flow that's great for northern populations because they're receiving more gene flow from farther south. Farther south, we're going to have more warm adapted trees. And so this asymmetric gene flow is going to take warm adapted genes north, pre-adapted alleles coming in. But what about the southern populations? Well, gene flow from the center south is going to exacerbate the effects of climate change because you're going to have colder adapted alleles coming down. So we get different things going on in different places. And this pattern of gene flow may accelerate the demise or inhibit the ability of more southern or milder populations to adapt. Now, the good news in all this, perhaps, is that we find, because of the high gene flow levels, that there's a lot of genetic variation in all of the populations. And when we look at a particular genetic SNP that's telling us something about adaptation, where you have two possible forms, most populations have most of that variation. So these numbers here tell you of 100% of the adaptive variation that we found in Sitka spruce, what percentage is missing in a given population. So 0 means they've got variation at all of those, in all those genes that we found were important. And you find most of the populations have most of that variation, and it's only when you get out to the edges, here the southernmost population missing 18%, and when we go to this northwesternmost population, Kodiak Island, that was very recently founded, we find it's missing about 40% of the variation. But the vast proportion of the range has most or all of the variation present in the other populations. And so how do you get different trees? Well, the frequencies differ. Up here, you're going to have the cold-adapted alleles at higher frequency. Down here, the warm-adapted. But what it means is you've got some warm-adapted alleles in the cold populations, and you've got some cold-adapted alleles in the warm populations. And that's good news, because it means on an evolutionary time scale, you've got the raw material in those populations to adapt. However, this is a very slow process, and this is going to take generations. And adaptation, through natural selection, requires mortality. It requires that a whole bunch of the trees do not survive, do not reproduce. And so, are we going to have a bunch of tree species going extinct? No. We have some at risk that are under threat from other things. But our widespread species, they're still, in all likelihood, going to be around. But our forests might not be very pretty. And this is a 2010 review of drought and heat-related dieback of forests around the world. And we're already seeing the kind of mortality that might result from these sorts of things. So, we could let nature take its course, but we're probably going to end up with pretty unhealthy forests, forests that aren't producing much in terms of wood and timber supply, forests that aren't sequestering much carbon, or maybe are sources of carbon, and forests that maybe aren't providing the best habitat for a lot of the things out there that we rely on forests to provide habitat for. So, should we be moving things around, or can we move things around? So now what I'd like to focus on is what our management options are. Again, back to our old cartoon. We've got populations at this lagging edge that we expect are going to be at high risk of being lost. And we should probably be monitoring those. And we should probably be making seed collections and putting seed in seed banks to capture that genetic variation. And we should be thinking about these populations as sources, seed sources, for farther north or farther into colder environments in the species range. In the proportion of the range that overlaps, of course, we still got the trees there, but they're in the wrong places. And we need to think about assisted migration of populations within that range. Then we have these new areas opening up. And they're opening up faster than trees can naturally migrate. And perhaps we should be thinking about moving some trees into those areas. And I'll be talking about each of those. So let's talk about those areas that overlap, where the historical range overlaps the projected future range. and we have an area that's been harvested and needs to be replanted, or maybe it has been disturbed for some other resource activity and it needs to be restored, and we need to plant something there. So perhaps we should be making our best guess about what is going to do well there in the future so that we have pre-adapted future forests. Now, there's a lot of questions around this. What age of trees do we target for optimizing that adaptation? Do we perhaps want to be mixing seed sources, just as we should be mixing species under the uncertain conditions that we're focusing? So here, this schematic depicts mixing two seed sources because there's a lot of uncertainty around what is going to happen. And this should buffer the stand over that possible range of future conditions. Now, if we get the single provenance right, we would expect probably higher yields from that, but there's a lot of uncertainty there, and this reflects a sort of portfolio effect, where by mixing seed sources, the worst outcome should be better under this scenario than under a single source scenario. Here I've depicted mixing more southern adapted and the more local individuals. So we've started on a large genomics project that is really focused on, can we use information about adaptation at the DNA level? And I have to say, even 5 years ago, we couldn't even contemplate doing something on this scale. But can we then take genomics tools combined with traditional methods of common garden experiments, and can we help inform what the genetic effects will be of assisted migration of populations, and how can we learn from the existing distribution of genetic variation to assist migration in a way that is reduces risks and increases the chance of having productive stands and good flow of timber and other ecosystem services in the future. So Andreas and I were fortunate enough to land a large-scale applied research project funding from Genome Canada, Genome BC, Genome Alberta, and a lot of co-funders, who I will mention at the end. So we put together a team. Andreas and I are co-PIs. And we put together people who are expert in genomics, in population genetics, social science. Deborah Davidson is here at U of A, and climate modeling. So we put together all these people to try to address this question. And so the simple justification for this goes as follows. We have seed source on public lands dictated by forest policy. And the success of adaptation, the degree of adaptation of the materials we plant affects reforestation success, the cost of reforestation, especially if you have to go back in, determines the growth, carbon sequestration, and finally, timber supply. So if we can inform this end of things, we could have a big impact. Here's an example from lodgepole pine, an economic analysis and an analysis, a projection of growth if we keep using local seed, which is the current thing that foresters do. We use relatively local seed sources. And with lodgepole pine, this is a middle-of-the-road, using a middle-of-the-road warming scenario. This is the change in productivity we expect to see for lodgepole pine. This is in BC. And we expect to see a slight increase in productivity in the short term on average, because we have some cold-limited sites. But we also expect to see some sites with decreasing productivity right from the get-go. But in the longer term, we expect to see productivity drop off between 10 and 35% if we continue to use local seed. We crunched some numbers based on BC and Alberta harvests. And if we can offset just 15% of this decline through assisted migration, that's worth about $200 million annually. So how are we doing this? Well, we're using the signature of natural selection in natural stands. and we are using a lot of stands. So we have seed from across the ranges of lodgepole pine and spruce. And when I say spruce, I mean white spruce, Engelmann spruce, and the hybrid zones. And we have large numbers of populations, 281 lodgepole pine, that's supposed to say 254, spruce. And that's a lot of populations, and that can tell us, we've selected these, Andreas, Tong Li, and others selected these to sample the full climatic range that's out there using their climatic model. So we have a really good distribution of seed. And I should say we've had tremendous support on getting the seed for this project. And this is a list of the companies and organizations in Alberta that have allowed us to use their seed for this project. And so there are people representing many of these organizations in the room, and I thank you very much for that contribution. And the list is equally long for BC. So that really let us get this project off to a fast start. So we are growing trees from those hundreds of populations for 2 species in a series of growth chamber experiments. Here are 3 of our treatments in pine. So we simulate different mean annual temperatures from 1 to 11. And we also have some treatments. The hot one includes also a well-watered and drought treatment. And we phenotype them for bud phenology, for growth, for cold hardiness, for drought hardiness. We expose them to heat stress. And we'll be looking at water use efficiency using stable carbon isotopes. We also have a set of materials from seed orchards in Alberta and BC. We have about 20 seed lots. And these represent the different seed zones that those come from, and also the dots represent the natural population samples. We'll be comparing those two. This experiment has about 6,000 trees in it, and so we can't grow it in a growth chamber. It's outside, but we're phenotyping those for many of the same things to see if the material that has been selectively bred is any different for these traits or these genes. Now, when we started out this project, our goal was to look at 10,000 genes. I was writing the proposal 2.5 years ago, and I wrote down 10,000 genes, and I thought it was crazy. Like, we're overselling. 10,000 genes, really? Can we look at 10,000 genes? We're looking at 30,000 genes, which is about the estimate of how many genes each of these species has. And it's the new advances in technology, the next generation sequencing, plus the ability to go in. These conifers have huge masses of DNA in their genome. They have 7 times as much genome as a human does, as much DNA in their genome as a human does. And we have to separate the wheat from the chaff. And the methods for doing that through what's called targeted gene capture have advanced hugely and become much less expensive. So we are now going after 30,000 genes. We identified those sequences through transcriptome sequencing. We extract RNA from seedlings grown under a range of conditions, and then we put those sequences together. And we were able to use draft whole genome sequences that just came available from white spruce, from York Bowman's group, and from loblolly pine, from David Neal's group. So we're really just riding a technological wave here where what we can do is changing by the month. So we're going after 30,000 genes, and we will sequence every one of those genes in about 750 trees per species. And that's a lot of trees, and it's a lot of DNA. I should point out the DNA here, or the RNA sequence here, if you printed it out on 8.5 by 11 paper, single spacing, 12-point font, you'd have a stack of paper 6.1 kilometers high. And it took us a few months to get those data. So it's absolutely mind-blowing. So now we're going to resequence those, and we are going to get about 500,000 single nucleotide polymorphisms. So I was talking about 35 SNPs here and 200 SNPs there. We'll be looking at about 500,000. And we'll be looking for those SNPs that show correlations with climate or that show correlations with those traits we care about. Timing of growth, growth rate, cold hardiness, drought hardiness. And the most promising of those we'll take ahead and we'll genotype those, and we're expecting to have a few tens of thousands of those. We'll go ahead and genotype those in total across the 2 species in about 12,000 trees. So it's just mind-blowing to me we're able to look at this much of the genome. But the real bottom line here is we have to bring this all together. We've got to look at the traits. We've got to look at the genes. We've got to look at the climates. And with Andreas and his group, and with Tong Li in our group, we've got tremendous expertise in spatial modeling. And so we'll be able to then look at a range of options for climate-based seed transfer and say, what are the genetic effects of this? How different are these two populations? If we mix them, do we get anything that's much different from just taking this other population? And how can we best design climate-based seed zones for the future that will manage that adaptive variation and better maintain a match between genotypes and sites? This is about the only data slide I have from this project. This is just some of our gene expression data. Here we have about 50 seedlings. Each row is one seedling here. We've got 100 genes that showed big differences in gene expression. And this is for lodgepole pine. We did this for both pine and spruce. And you can see different treatments here. Genes that were, each row is a gene. And if it's turned up in a given treatment, it's yellow. If it's turned down, it's red. And what you see is massive changes in gene regulation, gene expression, when we put them in different environments, cold, wet versus hot, dry. You see big differences here. Mild, wet conditions followed by heat stress. You see a very different transcriptome. And we found that there were 11,000 genes that changed in expression among these treatments for lodgepole pine and about 9,000 in spruce. And so I think what this reflects is just the complexity, the genomic complexity, but we can start getting at that because we can now look at so many genes simultaneously. Now this work, all large-scale Genome Canada funded projects require what's called a GELS component. GELS stands for Genomics-Related Ethical, Environmental, Economic, Legal, and Social Research. And that's where Deborah Davidson and Kevin Jones, Deborah's research associate, is here, and Rob Kozak and his crew at UBC. They are asking questions around different strategies for reforestation given climate change. What does the general public think of those strategies? What do professional foresters think? What do forest-dependent community residents think? And Deborah and Kevin are doing a policy analysis in each province. They're doing a policy regime analysis and interviewing all the key policymakers and experts in those areas. So we're putting together, we're not just doing a biological study, but we're trying to put that in the context of how do we deal with these forests, these on public lands, to sustain a timber supply into an uncertain future. Now, just this is a bit of an aside. We are spending millions of dollars on our study with lodgepole pine and spruce, which are the most economically important tree species. In BC, we have 50 tree species. And that's a lot of them. We don't know anything about the genetics of a lot of those species. And the same applies to all the other organisms out there that we don't know much about the genetics. I'm just asking the question here, for those species we can't put these kind of resources to, how similar are the patterns of adaptation across the same climatic gradient? So if the x-axis here is the climate, some climatic gradient, and the y-axis is some trait, do we expect that different species have the same kinds of patterns of variation? So Jordan Bemmels, a former technician in my group and undergrad, he did an analysis of information available from the literature on this, and this is just one of the graphs that he produced. But if you look at, we looked at all the tree species we could find common garden data in Western North America and looked at the declines that you find in a common garden based on provenance, mean annual temperature. He looked at a bunch of different climatic variables. The way that these data have been analyzed in the past is by latitude and longitude and elevation, because people didn't have good climate estimates. But we're putting them on a common climate basis, and there were 21 species in this, 21 studies, 16 species in this analysis. And what you see is that many of these species have very similar patterns. Now, the one with the very steep pattern is Sitka spruce, so we made a really good choice when we first started working on Sitka spruce because it's got such steep climbs. But my question here is, so if we don't know anything about genetic variation in a given species, should we start just migrating based on climate? And my argument would be yes. We have a lot of species with similar patterns here along this particular climate gradient. And if we start moving things, I mean short distances, but if we start moving things, let's say it's a species with no variation among populations. All the populations are the same, and we're moving them from warmer to cooler environments a bit. What's the downside of that? Not much. You're getting your seed from somewhere else. That's the downside. But really, if you're planting the trees anyways, not a big deal. If they have a steeper relationship than the average, like Sitka spruce, what's the downside? Well, at least you're helping get started on dealing with the problem. So I think we need to think about moving ahead with a generalized set of small movements for not very economically important species that we're using in restoration and things like that. Okay, finally, there has been a big debate about assisted migration. And I'd like to spend a little bit of time on that. 2 weeks ago, I was interviewed on CBC's The Current, and that was set up as a debate on the topic of assisted migration. I had the foresight to ask the CBC who my opponent would be, professor at McGill, Anthony Ricciardi. And we talked. I phoned him. I thought, let's talk, because I want this to be a discussion and information, education for the public. Let's talk about the issues, but let's not take polar sides, because I think we probably agree on a lot of things. And in fact, it turned out we agreed on almost everything. So it wasn't a debate, but you can listen to it. It's online. So anyways, I'm starting to get concerned about global warming. The sad thing is, I think that's New York. So, but anyways, so assisted migration has been called a bunch of different things. Managed translocation, assisted colonization. And it has been a hot topic in the literature. But most of the discussion has been about taking species from where they exist now and putting them somewhere far away where they don't currently exist. Now, I call that introduction of exotics, and I think that is a different matter. We're not talking about short distance movements, that's talking about long distance movements. There has been work, though, also on the short distance movements, and we call that assisted migration as well. So this shows the publications and the interest, the increasing interest. This is a review of the literature, which is mostly reviews. That's how scientists work. You review the reviews, and then you review the reviews of the reviews. So anyways, and you can see the number of publications. But it's interesting that if you look at the distribution of these, look how many of these papers were literature reviews or commentaries. So there was no new information added to the mix with those. It's people taking available information and using it to argue one side or the other. And it really does fall down to one side or the other in a lot of this literature. Here we have reasons for assisted migration in dark, reasons against assisted migration. This is another review of the reviews. So reasons for in dark, reasons against in light, and different categories of methods or reasoning. And what you see is these things are almost matched. Reasons for and against are matched almost the way through. So that's one interesting thing about this. But the other interesting thing is, look at the number of empirical studies. Wow. Lots of talking, not much doing. So some of the things people have said. So Mintier and Collins think we better get on assisted migration from a conservation standpoint. The debate over managed relocation reminds us that the metabolism of conservation must accelerate if we want to keep up with changes that are outpacing traditional ways of managing species and ecosystems. Okay, so we got to get on it. We're losing species, let's go. And then you've got Ricciardi, who I was on CBC with a couple of weeks ago, Anthony Ricciardi and Dan Simberloff. They're both invasive species biologists. They work in aquatic ecosystems where invasive species have wreaked havoc in many cases. And so they say assisted migration is ecological gambling and a planned invasion. And so they're viewing it as this long-distance introduction of exotics. And finally, Fazey and Fish are also against a techno-fix that does not address the root causes of extinction. That is a fair comment. However, our world is changing very quickly. And so we can argue this, but meanwhile, how many species will we let go extinct if we're thinking from a conservation standpoint? And meanwhile, from a forestry standpoint, how much decline in health do we want to see in our forest stands? Now, invasive species and long-distance introductions are a different matter. Foresters tend to be pretty comfortable with moving things around in some ways, or at least some foresters. And I think we need to remind ourselves that there are invasive species, situations that have been created by well-meaning foresters. New Zealand, we've got lodgepole pine, and we've got radiata pine naturalizing different parts of New Zealand, and lodgepole pine in particular has been a huge invasive problem. So we don't tend to think about our own species. But I think when we think about, so what's the difference between a reasonable assisted migration? Within the species range, really people aren't arguing too much. But that move outside of the species range, how do we define what's really an exotic introduction versus what's just helping the range migrate in the direction it's going anyways? And I think we need to take sort of an evolutionary and paleo-ecological perspective on that. And these maps were generated by Dave Roberts, who is a PhD student in the Department of Renewable Resources here, and he was collaborating with a student of mine. And this shows from 21,000 years before now until present day. Dave has backcasted where the climatic niches were located for Engelmann spruce in green, white spruce in blue, and then brown is both species present. And like all of our species up here, these species have migrated a long ways. And they migrated along with a lot of other species. So we have these regional floras that moved up after the last ice age from refugia. And if two species were in contact over that period, then introducing one of them to an ecosystem that contains the other now isn't really introducing an exotic, in my opinion. It's bringing two species into contact that have had contact in relatively recent ecological terms. Now, I'm going to give you an example from a conservation standpoint of a species that I think might be worth expanding the species range, that is assisting the northward migration of. And this species is whitebark pine. And if any of you are hiking enthusiasts, you will know it. And this is a species, here we have a Clark's nutcracker, which is the primary seed disperser for whitebark pine, pecking apart a cone. This is a book that I'm a co-author on, a conservation genetics textbook that's coming out later this month. And we chose this as the cover because it represents a relationship between two species, and it also represents one species that's in a huge amount of trouble. So whitebark pine was listed under the Canadian Species at Risk Act this past June or July after recommendations from Kasiwik to list it so it was listed as threatened. It has been listed in Alberta and BC as well. Now, this is the range of whitebark pine in the US. This is the current range of whitebark pine. This is the projected distribution of whitebark pine in climates in 2090 in the US. About 95% gone. This is a species that's got white pine blister rust, mountain pine beetles, fire exclusion, you name it. It is just a poster child for disaster. And so my PhD student, Sierra McLean, and I set up an experiment, and this is an assisted migration experiment. Someone wrote recently in a paper that this was the first assisted migration experiment for conservation purposes in Canada. And I don't know if that's true or not, but I don't know anything to the contrary. And so here's the existing distribution in pale gray of whitebark pine, very inaccurate distribution maps that we have for whitebark. The dark gray is actually the distribution of the climatic envelope now for white bark pine. Okay? So the area that we think has the right climate to support this species. Most of our species, you get a pretty good fit between their range map and their climatic envelope map for current times. But white bark, we've got this huge area in the North Coast Range that is currently predicted to be favorable, but also our climate change projections suggest that it will continue to be favorable for this species in the future. So what Sierra did is she established 9 test sites, 3 within the existing species range, 3 within the climatically favorable areas outside the range, and 3 outside of those. And planted seeds, planted about 18,000 seeds out there. And in fact, we lost one area entirely, one site entirely, but otherwise, the projections, the establishment was surprisingly good. We got about 30% germination and initial seedling establishment. We got a little over 20% survival after a few years. And the sites that were in these areas, these dark gray areas like this up at Haynes Junction here, and in this area, the seedlings did pretty well there, the seed and seedlings. So seemed to support, very early data, but seemed to support those projections. And so then the question becomes, should we move it? We can move it, but should we? And that's a societal question, as much as it is a biological question. So let's look at what some of those risks of action or inaction are. And so here we have this same distribution, and what's the risk of moving things in these different zones? Okay, well, let's talk about moving the range north. We are going to alter those ecosystems. If we move a species in there that's not already in there, it must be different, right? It's got another species. The effects of that may be consequential or may not be consequential depending on what your criteria are, but the chances of real species invasions are unlikely if we're just extending these ranges. So we need to think about whether we want different species present than have historically been there. Within the range, people worry about something called outbreeding depression. These are genetic incompatibilities. That's unlikely to be a problem for trees for reasons that I don't want to get into that are a little technical. But if our predictions are wrong, we risk maladaptation. Do we want to risk that? Well, what are the risks of doing nothing? So if we do nothing, we can expect productivity to drop from a timber standpoint. We can expect carbon sequestration and some other functions to decline. We expect to see a slow adaptive response. So we expect to see maladaptation as well. And down here, we expect to see the extirpation of populations and the loss of genetic diversity that is present in those populations. So, in conclusion, I suggest that we should be moving seed within the species range, and we should be thinking about what circumstances to expand species ranges under. And we should definitely be collecting seed and putting it in the bank from these sorts of populations now before we don't have that option. And I'd like to thank all the many organizations that have supported this research and lots of people who have contributed. And I'd particularly like to thank Genome Alberta and Alberta Innovates Biosolutions for their support of our Adaptree project. And finally, I'd like to thank the University of Alberta and for all of you being here today.